Welcome dear student on my channel. Today we'll see the test of urine sample for its normal as well as abnormal constituents. So this is about the urine sample. Now the aim is there to test the presence of urea as well as sugar as well as albumin and the bile salts in this urine sample. Now before going to do this test, we'll just see what is the urine. So urine is what? It is a liquid byproduct of metabolism in the humans as well as in the many other animals. Urine flows from the kidneys through the ureter to the urinary bladder. Urination results in the urine being excreted from the body through the urethra. Your kidney makes the urine by filtering the waste and the extra water from your blood. The waste is called as a urea. Your blood carries it to the kidney. So each and every drop of the blood is there which is going through the kidney where it gets ultrafiltrated. So the normal urine is there which contains the urea about 60 to 150 milligram per liter, that is 2%. Normal urine is there which is of the pale yellow color. So what we can observe in this, which is of the pale yellow color. It is due to the presence of the pigment. So normal urine is there which is of the pale yellow color, and that is why it is due to the presence of the pigment, that is a urochrome. This urine is there which contains the 95% of the water and the 5% of the solutes in the form of the sodium, potassium, magnesium and ammonia, etc., which present in the dissolved state. Many organic compounds are also present in this, like the urea is there, uric acid is there, then the creatinine is there, etc. are also present in this. In case of the abnormal urine, what we find in case of the abnormal constituents are there, which are in the urine, there the sugar is there, suppose albumin is there, bile pigments are there, blood is there, pus is there. If it comes in the urine, so that urine sample considered as a abnormal one. Now we'll perform the test for urea, for which the requirements are there. We require a test tube, we require the urine sample. So this one is a urine sample is there, okay. Along with that, we need the soybean flour. Another thing is that is about the phenolphthalein indicator. So again, repeat this. The requirement for this, that is a test tube, then the urine sample, then the soybean flour, then the phenolphthalein indicator. Now we'll follow the procedure for the same. Instead of soybean flour, you can use the gram flour also, which is as a source of the urease enzyme. Now we'll perform the test. First we'll take the test tube. In the test tube, approximately 2 ml of the urine sample. So here the 1/4 of the test tube I will fill with the urine sample. So 1/4 of the test tube is taken here. In this we'll add a pinch of the soybean flour. So here the pinch of the soybean flour I will add in this and then shake it well. So like that here we'll shake this. So what I have taken here, 2 ml of the urine sample plus pinch of the soybean flour. Now in this I will add the few drops of the phenolphthalein indicator. Few drops means 1 or 2 drops of the phenolphthalein indicator. So here this I will take the dropper and in this will add 1 or 2 drops of the phenolphthalein indicator. Then we'll just shake it and we'll see the result, what result we are getting in this. We'll keep it for the few minutes. Okay, after few minutes we'll get the result. We'll repeat the procedure here. We'll take the test tube. In that we'll take approximately 2 ml of the urine sample, then pinch of the soybean flour, then we'll shake it like that, and then after that we'll add few drops of the phenolphthalein indicator, and then we'll keep it for the few minutes. Now we'll observe the result after few minutes. After few minutes we got this, the color. So here the observation is that the mixture turned pink. Conclusion is that the urea is present in the given urine sample. So what we added in this? In the urine sample we have added the soybean flour. So soya provides ureas which catalyze the urea into ammonia and the carbon dioxide. Ammonia being alkaline reacts with the phenolphthalein and develop this pink color. So this pink color indicates that urea is present in the urine sample. Now urea is there, which is a nitrogenous waste product. It should get excreted from the body. So during the ultrafiltration, you already learned in the lesson excretion osmoregulation, in that the nitrogenous waste product should excrete from the body, and that comes out through the kidney in the form of the urine. So this presence of the urea in the urine sample, which indicates that the normal urine. Suppose the amount of the urea in the urine sample, if it is more than 20 to 40 milligram per 100 ml of the plasma, is called as a uremia. So uremia is there, which indicates the disease of urinary tract. Okay, so this is what we have performed, that is about the test for the urea. So this is a positive result what we get in this urine sample. Requirement for the test for bile salts, that is Hess sulfur powder test, for which the requirement, that is a sulfur powder. Then we require the urine sample. Then we have taken the plain water and then the test tube. So these are the requirements are there for the urine sample test. So we required two test tubes. In one test tube we'll take the urine sample, and in the another test tube we'll take the plain water. Now here this is about the bile salts. So bile salts are one of the primary component of bile. Bile is a greenish yellow fluid which is made by the liver and stored in our gallbladder. Bile salts help with the digestion of fats in our bodies. They also help us to absorb the fat soluble vitamin like the vitamin A, vitamin D, vitamin E and K. The bile salts are not normally found in the detectable quantity in the urine. More than 90% of the bile salts are actively reabsorbed by the body. The liver normally clears the 20 gram of the bile salts from the blood each and every day. Due to the liver infections like the jaundice, the bile salts are produced in much larger quantity. The salts are then excreted as a part of the urine, and so that we can detect that the person is suffering from that jaundice. Now here we'll perform the test for which the requirements are there. Just now I said the urine sample, then the plain water, then the sulfur powder and the two test tubes. Now we'll perform one by one. We'll take the approximately half test tube of the urine sample. Now simply we'll sprinkle this sulfur powder on the surface of this. So what we observe here in this, the sulfur powder sinks down. This is about the urine sample what we are testing in this. You can just check it, how this sulfur powder is going down. So slowly all whatever the particles are there about the sulfur powder which are going down, so this is the because the sulfur powder which sinks rapidly to the bottom in this urine sample. This is because of what? The bile salt present in this urine sample, which reduce the surface tension of the urine so that the sulfur powder sinks down. And this condition we don't find in case of the normal water or in case of the normal urine sample. So in this is about what? About the sample of urine which is abnormal. In the second test tube, I'll take the plain water. So half test tube, same way. I'll sprinkle the sulfur powder on this. Same quantity of the sulfur powder. So what I find here, nothing is going down, no particles. Even after the shaking also, this is not going down. So you can see the difference between this both. Now here in this, the sulfur powder sinks down, and in this the sulfur powder which is a floating. So in this test tube, whatever the urine sample is there, that urine sample considered as a abnormal one. It means it shows the presence of the bile salt. Bile salt should not get excreted from the body. It should get reabsorbed. As it gets excreted from the body, it means there is an infection in your liver, and that leads to the excretion of the bile through the kidney. This is about the plain water. So the bile salt is absent in this, and that's why the sulfur powder which is floating on the surface. So this is what we have performed, that is about the bile salt. Common bile salts are there, which are the sodium glycocholate, and second one is that is about the sodium taurocholate. So these are the two different bile salts are there. The next test what we are going to perform, that is for the test for the albumin, for which the requirements, we have taken the urine sample, then the nitric acid, which is a concentrated nitric acid, and the test tube. So these are the requirements for the test for albumin. Before doing the test, albumin means what? Albumin is a protein. Albumin is one of the protein found in the blood. Albumin is one of the protein which is found in the blood. It is made by our liver. Albumin circulates through your blood to help your body maintain fluid balance. Also, it helps to prevent the blood vessels from leaking too much. Albumin also has a role in repairing tissue and helping the body grow, while transporting vital hormones and nutrients around. Normally, during the urine formation, these large molecules of the albumin never escapes ultrafiltration. If presence of the albumin is detected, then it indicates the damage to the nephron, as in nephritis or the urinary tract infection. So this is about what? About the albumin, that is a protein which is present in the body. It should remain in the body, it should not get excrete. As it gets excrete, it means there is a problem in the kidney, or maybe a urinary tract, or either the person is taking the high protein diet, or sometimes in case of the pregnant woman also we find the presence of the albumin in the urine sample, that is considered as a abnormal one. Now we'll perform the test. For the test, just now I said we require the urine sample, we require the concentrated nitric acid, and we require the test tube. So first what we'll do, we'll take the approximately 1/4 of the test tube of the nitric acid. It is a concentrated nitric acid, so you have to be careful while handling it. I'll repeat again. This is a concentrated nitric acid, so you have to hold the test tube carefully. Now this is approximately we have taken the nitric acid in the test tube. Approximately it is 2 ml. Now same quantity of the urine sample, I will pour it in this. We have to hold the test tube at 45 degree, and then we have to release this urine sample. We have to hold the test tube at 45 degree, and then slowly we have to release this urine sample, same quantity. You have to touch the dropper from the inner side of the test tube, and slowly you have to release this. Same quantity you have to add in this. Slowly you have to release. What we find here in this, the white cloudy ring at the junction of two liquid. This white cloudy ring is there, which indicates the presence of albumin in this urine sample. You can clearly observe this, the white cloudy ring. This is a floating in between the two liquid. Now we'll perform the test for sugar. Now this is about a urine sample which is taken for the checking the sugar level in this urine sample. Second, we require the Benedict reagent and then the test tube holder as well as a spirit lamp. Okay, so these are the requirements are there for the test for sugar. Now sugar present in the blood. The level of sugar rises in the blood because of why? The hormone insulin, which is secreted by the pancreas. Suppose there is a defect in the pancreas which secrete the insulin less, as a result the sugar level in the blood get rises. So sugar level in the blood which is controlled by the hormone insulin. So deficiency of the insulin hormone, that is from the pancreas, which leads to the diabetes. Now we'll perform this test. We'll repeat again. We require the urine sample, we require the spirit lamp, we require the Benedict reagent, as well as we require the test tube and the test tube holder. So in this test tube, first I will take the 2 ml of the urine sample and then we'll add 1 ml of the Benedict reagent in this. So 2 ml approximately I will take the 1/4 of the test tube of the urine sample, and now in this I will add 1 ml of the Benedict reagent, so 1 ml of Benedict reagent, and then we have to boil this over a spirit lamp. Now here we'll hold this. So this you have to boil for 2 minutes. Now suppose the color of this mixture will remain blue, then it indicates that the sugar is absent, glucose is absent. And suppose this color changes to the yellow, then it indicates that it is having the 0.5% of the glucose, and suppose it changed to the orange color, then it indicates that it is having the 1% of the glucose. And suppose if it is a brick red color, then it indicates that it is having 1.5% of the glucose. So here we can observe that it is a somewhat the orange to brick red color precipitate is there. Okay, so you don't have to hold continuously, slowly you have to heat it, and then we get the result that is of brick red color. Now this whatever the orange to brick red color precipitate is there in this urine sample, this indicates that it is the diabetic patient's urine sample is there. Okay, the person is suffering from the diabetes. It means the glucose level in the blood is more. So sugar present in the urine sample, the glucose reduces the alkaline CuSO4. The glucose in this urine sample, which is reduce the alkaline CuSO4 present in the Benedict reagent. What we added in this, the Benedict reagent. So this Benedict reagent to the cuprous oxide giving the brick red precipitate. So here we get the orange red to brick red color precipitate. Now in case of the normal human, urine does not contain sugar or does not contain the glucose in the urine. But the urine of the diabetic patient, if it's the urine of the diabetic patient, so here it shows a sugar. Now condition is called what, the glucosuria. Suppose it contains a sugar, then the condition is called as a glucosuria. It is due to the deficiency of the insulin hormone, which is secreted by the pancreas. So insulin secreted by pancreas, and suppose this level of the insulin is less, then automatically the level of the glucose in the blood will increases, because the insulin controls the level of the glucose in the blood. Due to the deficiency of the insulin, blood glucose level rises and the glucose finds its way into the urine, and that condition is called as a diabetes mellitus. In case of the abnormal decrease of the sugar level in the blood is called as a hypoglycemia. So the sugar level also can get reduced, level can go down. So that forms what? Hypoglycemia. A blood sugar level less than 140 mg per dl is normal, while a reading of the more than 200 mg per dl after 2 hours indicates that the diabetes. A reading between 140 to 199 mg per dl indicates that pre-diabetic patient, means that is in the border level. So this is about what? About the test for the sugar. So here in this result, what we got? The positive result. So presence of the whatever the test we have performed, presence of the urea, that is considered as a normal one, but the presence of the sugar, presence of the bile, presence of the albumin, this all the urine samples are considered as a abnormal. Okay, so we have performed this to understand about the normal and the abnormal content in the urine sample. Thank you. If you like my channel, subscribe and share with your friends. Thank you.